And thank you very much, Matt, for agreeing to give the Build a Cell seminar, and it's all yours.
All right. Hi, everyone. I'm Matt Good. I'm an assistant professor in cell and developmental biology and bioengineering at the University of Pennsylvania. I want to thank Kate for the invitation to speak. It's certainly a thrill to be here to present our work in the Build a Cell seminar series. And today what I want to discuss is our research on building designer protein-based condensates inside living cells for modular control of cell behavior. So my group has been interested in building designer biochemical compartments inside living cells, starting from polymer building blocks that can be genetically encoded. And we'd like to use these or leverage these to then assemble synthetic compartments inside cells that we can use to regulate cell decision-making. And really our goal was kind of twofold. One, we wanted to build a system that we could biochemically characterize ex vivo, so we'd have some biophysical understanding of the components. And then we also wanted to build a protein-based system, one that might quickly respond to temporal initiation, something much faster than like a traditional gene circuit that's based on transcription and translation. And we're also interested, and others have done this as well, in a system that might allow multiple cycles of input-output responses. And really what we're talking about here, sorry for the delay, is the attempt to build a synthetic organelle that could either serve one of two functions, either to sequester, insulate components inside a cell to shut down activity, or to concentrate components in a bioreactor scheme to accelerate reactions. So there's a number of platforms one could consider for trying to engineer these compartments inside of cells. And there's been a number of previous efforts that have leveraged existing bilayer compartments in cells, for example, to accelerate metabolic reactions by anchoring enzymatic cascades to mitochondria to peroxisomes. Alternatively, one could build synthetic vesicle systems from scratch, and my lab has done so for a number of alternative amphiphile systems. Now, in practice, these really have most utility ex vivo. They're not easy to construct in living cells. It's quite difficult to engineer lipid metabolism. And so these would really have to be fed continuously to cells to be able to modulate them. So I mentioned in the previous slide, our interest was really in genetic encoding of organelle assembly, and this pushed us towards protein-based systems. So what I'm showing here on the upper right, this could include nanocompartments such as encapsulins and protein nanocages, which are a very attractive approach. But what we really got excited about was thinking about how to assemble mesoscale protein and RNA condensates. And these are the compartments I'm going to discuss today and how we're using them to partition enzymes in cells. So in our design, we took inspiration from natural membraneless organelle systems. You guys all know this, but briefly, the cell contains both compartments bounded by lipid bilayer membranes and also those that assemble that are protein-rich bodies that we term membraneless organelles. This includes cytosolic and nuclear bodies such as the nucleolus. And I like to highlight these large embryo-specific condensates like germ granules and the Balbiani body. And these subcellular assemblies, they function as selective biochemical niches. Now, what's interesting is their assemblies are thought to form from the coacervation of nanoscale proteins and RNAs into these micron length scale biomolecular condensates. So a fundamental principle at play here is that disordered polypeptides, polymers, when above their critical concentration, self-assemble via weak multivalent interaction into these mesoscale condensates. I'm just showing you a typical phase plot, and above this line, polymer is miscible and below is condensed. So one of the ways we understand this is by purifying recombinant proteins in vitro, which will undergo liquid-liquid phase separation, such that in a turbidity assay looking at the large scale, you'll generate turbid solutions, and under the microscope, see the assembly of these micron length scale protein condensates. So a little bit more about the parameters of the system. Again, showing you a phase plot here for a disordered protein that displays upper critical solution temperature behavior. Above this line, the protein is miscible, and if we follow a tie line, we can watch this protein transition as you increase concentration to a condensed phase, and then finally an inversion of this continuous phase. And what we got excited about this system, one reason we did is, we could build these ex vivo, we could characterize them, but also we can increase their size and thus potentially reaction flux by raising the concentration above the saturation concentration. Another interesting property of these systems, one that is a little scary for engineering, is that they're highly dynamic. Although certainly some of the material properties of different IDRs will end up being in a more gel-like state. Most often they display liquid-like behaviors, including rapid fusion of droplets and this dynamic recovery from photobleaching. So one major question at the beginning of our studies was, which disordered protein scaffold to use. There's certainly many choices for building condensates, and which would we select for constructing synthetic organelles from the bottom up. And so we chose the RGG domain from LAF1. This is a model disordered protein our group has been characterizing and engineering, which normally forms structures called P granules or germ granules in embryonic cells of, in this case, C. elegans. And these help partition RNAs specifically into the germ lineage early in development. In particular, this 168 amino acid disordered N-terminal region, this RGG domain is necessary, the Brangwyn lab showed is necessary and sufficient for liquid droplet formation in vitro. And in a study we published last year with the Mittal lab, we used computational modeling and in vitro reconstitution of purified proteins to identify sequence rules governing condensation of the LAF1 RGG domain. And so I want to tell you about a few features that we discovered. From the modeling, it became clear there was this 10 amino acid patch near the N-terminus, which is clustered in the coarse grain models and is required for this higher transition temperature. In addition, we demonstrated the importance of arginine-tyrosine interactions, showing you them along the linear sequence here. They're well distributed. And the reason we know they're important, if you look at turbidity assays, so absorbance is a function of temperature, wild type is in black. If we mutate all the arginines to lysine, then we completely destroy the ability of this polymer to condense. And a third feature that was, by the way, I should mention, this is very similar. These residues are also important for PhET family members like FUS, the FUS-LC domain, although these tend to be clustered in the FUS protein, whereas they're well distributed here. So the third feature I want to mention is that we were able to do sequence redesign from the coarse grain modeling that really had a predictive tuning of critical temperature that we were able to validate in vitro. And so without changing the composition of the sequence, which is shown here, just highlighting, in this case, anionic and cationic residues, the Mittal lab identified these shuffled, same composition, but shuffled sequences where charge was clustered to the termini, and these had a profound effect on the transition temperature. Now, under these conditions, the single RG domain transitions at about 15 degrees, and then the shuffled D, which is the shuffled sequence while maintaining this short hydrophobic patch, shows a transition temperature of around 50 degrees Celsius. So this was really kind of a dramatic prediction of or tuning of critical temperature. And so I want to just mention that collectively, this study and certainly those of others from the past year or two, we now understand a number of types of interactions that drive homotypic polymer-polymer interactions and lead to condensation. So one other thing I want to mention is now with this knowledge of our ability to weaken or enhance the critical concentration, critical temperature for phase separation, we of course wanted to know, were we changing the material properties of these condensates as well? And intriguingly, all the mutants that I just mentioned to you, whether or not they were weakened, like tyrosine phenylalanine or deletion of this hydrophobic portion, or the shuffles that have this dramatic increase in critical temperature, they all retain their liquid-like properties. And that data is for measured by droplet fusion relaxation times here in the middle, or on the right by photobleaching. All right, so switching gears briefly, I also just want to describe previous results that demonstrate this RGG domain is an excellent IDP platform for controllable condensation in vitro. So a postdoc, Ben Schuster, took the approach of increasing RGG domain valency and showing with these turbidity assays a transition from 15-degree transition temperature to 37 and 50 by switching from single tandem or triple RGG domain. He initially also used enzymatic regulation to, in real time, reduce the valency to dissolve these condensates or to remove a tag to trigger de novo droplet formation. And his strategy was to engineer in between these linked domains, proteage cleavage sites, to enable this modulation of valency and saturation concentration. And further, he showed that these condensates have selective permeability. They're functioning like, what we would expect a membrane-bound or membraneless organelle. He showed that they have a size dependence, a mesh size dependence, but in spite of this mesh size, they're definitely sufficiently permeable enough to allow diffusion in and out of proteins such as RFP and GFP. But that's not enrichment, right? That just allows diffusion inside the condensate. And so to get selective recruitment to concentrate cargoes inside, Ben added short peptide tags, synzip interaction motifs to scaffold and client, and I'll come back to this in a minute as I talk to you about some of our new data. And then more recently, a grad student, Ellen Reed, and a postdoc, Mikhail Garabedian, they embedded light-based regulatory controls in the RGG sequence to create optically responsive polymers and demonstrate their photo switching inside these cell-like compartments in vitro. And so instead of requiring enzymatic cleavage, we're able to do this with photo cleavage or photo linking of the domain. So on the left side, showing you a system called SPLIT. This responds to photo cleavage of an encoded focal tag to promote condensation over a time course of minutes. This is a non-illuminated and illuminated sample right next to one another and showing condensation inside these cell size water and oil emulsions. And on the right is a system based on real-time modulation of IDP valency to generate either tandem or triple RGG by clustering them together to jump across the phase boundary. And this leads to inducible condensation, again, on the order of tens of seconds to minutes. Okay, so briefly to just summarize some of the parts and principles that we learned from these early studies. We understand sequence determinants guiding LAF1's condensation, the RGG domain. We can tune critical concentration and temperature but through RGG valency, demonstrated some understanding of permeability and client recruitment, and have generated regulatory handles that allow controlled assembly and disassembly through enzymatic and optical means. And so for the rest of the talk, really what was driving this project was trying to integrate this knowledge to build synthetic membraneless organelles that would be expressed in cells and that we could use to control the flow of information and thus drive cellular decision-making. Okay, so this new study, this is a paper that's just been accepted. I'll explain how we deployed these RGG-based condensates to insulate native enzymes and enable modular control of cell behavior. So there's a number of excellent existing synthetic condensate platforms. We could spend a long time talking about them. I'm not going to review them here, but one of the kind of key points is that we sought to engineer a platform to achieve a number of functions not available in these off-the-shelf systems. And so what kind of our goals are things that are likely generalizable to other IDR systems, but we had to first implement them in our own kind of synthetic organelle approach. And so a major aim was to generate IDR variants that had very low saturation concentrations so that when the protein's expressed in a cell, we can achieve a vast majority of its partitioning into the condensate This is really important for the client recruitment side. And then the second note, we wanted to selectively and efficiently recruit native clients. And we tested a number of short interaction tags and used CRISPR to knock them into the C-terminus of endogenous gene loci. And then because this is a protein switch and we're looking for kind of rapid control, we wanted to achieve inducible client recruitment and inducible controlled release from these compartments. And all of this is for the purpose of controlling the activity state of a cell, using these synthetic organelles to target pathways of interest. Okay, so kind of our first step was just to repeat these tests of temperature stability, which I mentioned to you in vitro as a function of RGG domain valency. And instead to do this inside living cells and as a test bed, we used the industrially relevant model organism budding yeast. And so again, here's the in vitro data, single, tandem, and triple RGG domains. So we repeated this in cells, adding a GFP tag so that we could track the assembly with imaging. And I'm showing you those three variants as a function of three different temperatures. We use a cherry temp so that we can grow these cells at these different temperatures. And so what you can see is that the single RGG domain does not condense at room temp. We get very, very small condensates by cooling it to 4 degrees. The tandem RGG with GFP nicely forms condensates at 25, but these are largely dissolved at 37 and absent at 42 Celsius. And the triple nicely forms condensates essentially at all three of these temperatures. And so this really seems to match our turbidity measurements in vitro. And importantly, the triple RGG condensates are still highly dynamic and liquid-like as shown from photobleaching inside of the cell. And so in large part, we expect a lot of these principles, this biophysical characterization ex vivo to then hold at least in these cells here. So our second goal was to characterize protein interaction tags for selective recruitment of client with the goal of trying to achieve 90% sequestration, right? We set this mark because we wanted to have some kind of functional knockdown of the pool of a protein inside of a cell. And I want to walk you through a rationale for selecting coil-coil link scaffolding client versus just an IDR. And so one common strategy for client recruitment to IDB condensates, if you've followed this in papers published over the last 2 or 3 years, has been to have one scaffold with an IDR to generate condensates, and then to also tag a client of interest with that IDR sequence, such that this has the benefit of mediating recruitment to the condensates when the condensates are formed, rather than simply a one-to-one interaction between scaffold and client. However, a lot of these interactions are much weaker. We were concerned this would limit our ability to sequester, to try to get 90% of native clients sequestered in a cell. And we were also concerned that tagging clients with large IDRs would potentially lead to loss of native function. So previously, we actually tested this in vitro. Ben Schuster tested this. And what he found was by tagging a client, in this case, the cargo is just red fluorescent protein with a single RGG domain. So this would be the IDR kind of enrichment approach. And then looking at recruitment to preformed condensates, that it's actually quite weak. It's just modest enrichment. And I know it's a little small on the screen, but there's actually enrichment on the surface of the condensates. And this is in stark contrast to using, for example, these short coil-coil domains. These are synzip pairs designed by the Keating lab. And he was able to show this is all normalized to the max, which is a cargo that has 2 full IDR domains. It's able to actually condense on its own based on the concentrations we're using here. And so we can achieve almost that level of recruitment just using these short coil-coil tags, whereas the single IDR domain on its own, again, is much weaker at recruiting clients to these condensates in vitro. Furthermore, a postdoc, Mikhail, repeated these experiments in cells and found very similar results that we could get, 20 to 50-fold higher enrichment using coil pairs compared to just using the IDR sequence. And so thus we decided, for this project, at least for generating functional knockdowns of native pathways for this focus, we would really need to tag the scaffolding client with cognate high affinity coil coils. All right, so for testing the coil coils, we characterized synzip tags and thermally responsive coil coils. These are a sub-region of the bacterial thermal sensor, TLPA. On the top of these images, you can see the scaffold expression in green, clients in the middle, and the merge is on the bottom. And so what you can see is the client's very strongly recruited to both types of, to condensates using both types of tags, either the synzip tags or the TLPA tags, which we call TSCA, TSCC-A and B. That interaction is evidenced from the merge. I'll also show you the quantitation in a second. I do want to also point out that the thermocoil tag scaffold leads to formation of largely just one large condensate per cell. This is likely because of some modest homotonic interaction among this TLPA subcoil that leads to elevated rates of fusion. Nevertheless, the scaffold in client partitioning is effective in both cases, but highly effective using the cognate thermocoil pairs. So if we calculate the fraction of scaffold inside these condensates, it's over 95% when using the RGG triple GFP tag to TSCC-A. And because of this, because we have such a high partitioning of scaffold, we also achieve over 90% partitioning of an exogenously expressed client. And so this pair really gave us, the belief that we might actually be able to target native clients inside of the cell. Coming back to the question about saturation concentration, we characterize it directly to demonstrate the importance of valency and this TLPA tag. So these are the variants we tested for expression in cells, and what we can measure is the amount of signal in the cytosol versus the total signal inside the cell. And this is a line that basically describes the phase boundary. And so the single RGG domain, although it's expressed, these are from a gal promoter at over 5 micromolar or around 5 micromolar. This does not condense. Whereas when we look at the tandem, this one in red, the tandem with the synzip tag, it's expressed around 2 to 4 micromolar. It indeed does condense, and that critical concentration is around 2 micromolar. And then if we just switch to this log scale, this log plot on the right, so you can see it better, switching from the tandem to the triple further lowers the critical concentration. And then really, we get a big jump from using the TLPA thermocoils rather than the synzip 1 coil. And so the end result is that these dots in yellow, the average is a critical concentration of about 30 nanomolar compared to 1 micromolar expression in the cell, which is why we were able to achieve over 95% partitioning of the scaffold. Okay, so to test the ability of these condensates to sequester and functionally insulate native clients in the cell, we targeted a number of enzymes that control both sides of the cell cycle control system. For the proliferation side, we targeted the GAF-CDC24, and for the cell division side, we targeted the kinase CDC5. And the idea here is conceptually that normal flux through the pathway, for example, from CDC24 to activate the GTPase CDC42, would be blocked by sequestering this component inside of the synthetic condensate. So we genomically integrated the native gene into the native gene locus of fluorophore in this portion of the thermocoil tag. The first thing we wanted to look at is whether we would be able to disrupt native localization. So normally CDC24 is localized to polarized sites of growth and to the bud neck, and you can see that in these images. And CDC5, the kinase CDC5, is localized to spinal pole body. So this is these are tagged, and they're functioning normally. The cells grow normally, and these are their normal subcellular kind of sites of anchoring in the cell. And then when we express the condensates, the enzymes are displaced from their native subcellular localizations and sequestered to our organelles, which again you can see by the merge. And if we quantify the fraction of this native client that's partitioned to the condensates, we get about 85% of the total pool of the native enzyme which means like a 5 to 6-fold drop in cytosolic availability. So this seems to be working pretty well. So what about functionally? So, what happens if we sequester these native clients? Does this functionally insulate the enzyme from its partners? It's certainly possible that its substrate might still diffuse into the condensate and we wouldn't achieve a functional knockdown. So I'm showing here a spot dilution assay as a measure of cell growth. And I've got 2 columns on plates that are don't express the condensates, and then on plates that induce the expression of the condensates. This is done for CDC24 and 5. And each of these rows is a 5-fold spot dilution of the concentration or number of cells in the previous spot. And so what I hope you can see is that under normal conditions, when condensates are expressed, everybody grows normally. There appear to be no differences. Then if we look when condensates are present, we have the 2 controls, which are just the tag scaffold and just the tag client, but not together. And they grow normally. But then when we put them together, then we get this huge drop down in cell growth. And so it really looks like these are blocking the cell cycle control system. Further evidence for that is from looking at the percent of cells that are arrested or unbudded. Again, the controls look like normal, but when we put them together, the tagged scaffold and client, we get this strong arrest, as well as a 10-fold drop in proliferation rates in liquid culture. This is on plates, and this is in liquid culture. So it really looks like we can block the cell cycle control system, either these enzymes, but to further ensure this strategy is generalizable, we also targeted other factors, which I'll tell you about. One is the formin BNR1 that controls formation of actin cables during cell polarization. And so as expected, we can get around 80% sequestration of this formin. And the expectation then was that if we block the formin, we're going to block formation of actin cables. That's shown here on the right when we express the condensates. And indeed, we see this loss of both the cables and this overall polarization of the cell. And that's also quantified here on the right. So the take-home point is that these synthetic organelles and the tagging of native enzymes is a highly effective strategy for switching on or off a pathway of interest. And we suspect that most cytosolic nuclear or plasma membrane anchored enzymes that are expressed, let's say, below hundreds of nanomolar range could be effectively sequestered using this type of a platform. So because we set out to use these organelles as a protein-based switch, we needed to build from this platform and add regulatory handles that would allow rapid real-time control of client sequestration and controlled release. And so I'll just in one slide tell you about an induced client recruitment strategy. We followed kind of the same tagging strategies before, except instead of using coiled coils, we used FRB and FKBP domains that will dimerize in the presence of rapamycin. Here's a live movie showing relocalization of CDC24 away from its target sites to the condensates in real time, and quantitation of that, which shows that we can get about 50% of the client recruited to these synthetic condensates in about 10 minutes. And importantly, this induced client sequestration has a strong functional effect. So again, it completely shuts down growth as shown in these spot dilution assays. All right, so for controlled release, we're building these condensates. We're kind of constitutively recruiting clients. How do we release them in real time? We tested 2 strategies. One was a thermal strategy that would be reversible, and the second was an optogenetic strategy that would be irreversible. And so for the thermal strategy, we're relying on these thermally responsive coils that have been characterized previously, including in other labs like the Shapiro lab. And so as you raise the temperature from 37 to 40 or 42 degrees, you start to melt this interaction. And for the optogenetic strategy, we decided to implement this photocleavable protein focal, actually the 2nd generation one from the Campbell lab, between the interaction tag, thus to release the client of interest with only about a 17 amino acid scar added on to the end. Okay, so our experimental setup was to induce expression of the scaffold transcriptionally. That's the first phase. So that we'd form these condensates and sequester the targeted client. And then phenotypically, we can measure, for example, for cell cycle, what % of cells are arrested. And then in the second phase, to reverse this arrest by forcing client release. And so indeed, we see that cells, I'm going to show you this starting in black, we're growing the cells at 25 degrees. We're beginning induction of the condensates. And these are forming and beginning to sequester CDC24, which then leads to cell cycle arrest. At 6 hours after the induction, we then thermally reverse the system at 37 in yellow or at 42 degrees in red. This leads to, not rapid, but certainly under the time course of this experiment, reversal of this phenotype, the cell cycle arrest. What's important is that these temperatures, although, you can't grow cells forever at these temperatures, yeast actually do not mind growing at 37 degrees. And so you can actually maintain this reversal of client recruitment overnight just by keeping them at these temperatures. So at 25 degrees, they are fully arrested. And if we keep heating them, it's not possible for the client to be sequestered because we've melted that interaction. And this is just showing the fraction of client released after this period. It's actually only about a 30% release from the heating to 42 degrees, but this is enough to a sufficient amount of client release to reverse the arrest phenotype. Okay, so to test irreversible light-mediated client release, we fused photocleavable protein focal 2F to either the scaffold or client in between the thermocoil tags. For this experiment, I'm going to show it tagged to the scaffold. And what we wanted to test was how quickly and to what extent client could be released, again, using the GEF CDC24 as the test case. So we pulse cells in these 10-second intervals of light, and we're showing the fraction of this enzyme localized to the condensates, and you can see that we get a halftime of a little less than 2 minutes. And this could almost certainly be accelerated by the illumination strategy, but we were able to release about 60% of what's sequestered here over the course of 5 minutes, essentially. So what about cellular activity state? Is this release able to restore the cell cycle? So using a similar experimental setup to what I just showed you for the thermal release, we induce scaffold expression, and then we're going to reverse this at 4 hours after induction using a 10-minute pulse of light. And what was exciting is that we observe a near complete switching of the cells back to normal growth and reverse of this arrest. So the kinetics, of course, are much slower. We release the client within 2 to 5 minutes, and then it takes the cells about 2 hours to restore to 2 to 4 hours to restore back to normal growth. And this is maintained, although you can see that it's starting to tilt back up. And so, of course, as new protein gets expressed, then these would have to be pulsed again, or they'll be recruited back to the condensate. Finally, to demonstrate that we can cycle through multiple repeats of client sequestration and release, we subjected the cells to a cyclical pattern of temperature switching. And indeed, we observe switching between 4 states. So starting from 50% of the cells budded, we're able to send them to an arrested state, raise the temperature, bring them back up to a growing state, and then cool the temperature again to bring them back to arrested. So it looks like we can get cyclical control of client release and cell cycle behavior. Okay, so in this last section, I want to just show you that this toolkit can be used in a variety of cell types, including mammalian cells. So to demonstrate the generalizability of our platform and approach, we form condensates in mammalian U2S cells and used CRISPR tagging of the C-terminus of native gene loci for a variety of enzymes. We then transfected a plasmid expressing the scaffold to form condensates, very similar scaffold to what I showed you in yeast, but recoded. We can efficiently then sequester enzymes like ERK1 in mammalian cells. So, first I'll show you a few controls. We tested targeting of the native GTPase RAC1, and we can show on the upper panel that we can get very efficient client recruitment, sequestration of RAC1 if we use the improper coil pair so that we still form condensates, but it doesn't have the the cognate pair, then the client's not recruited. This really shows the specificity of the interaction. That data is shown here on the right, so we can achieve about 20-fold enrichment of condensate over background using the cognate pairs relative to the non-matching pair. And this seems pretty efficient on the whole. So starting from, this is fraction of protein inside the cell that's localized to the condensate. So we're starting from about 80% of scaffold partitioning. These condensates were able to sequester a little more than, or right around half of RAC1. The GTPA is RAC1 and the kinase ERK1. And what I think is important is that as with the yeast system, the cells that show higher expression above the saturation concentration, they show a higher fractional client sequestration. And so we're of course also doing this with lentivirus and integration into cells, but I want to just show you that this is feasible with transient transfection as well. So we're in the process of testing the functional impacts of enzyme sequestration. For example, we're trying to characterize the effect of insulating ERK1 in terms of its phosphorylation or nuclear shuttling. But one piece of data I can show you now is for the polarity protein PAR6 and targeting it to our organelle. So normally this is a protein that's polarized to the plasma membrane. When we express condensates in the cells, then this strips it off the plasma membrane. That's just quantified here on the right. In preliminary data that I'm not showing you here, my student Wentao is working on this, has observed that after we sequester PAR6, then the cells seem to lose adhesiveness. And this is one predicted effect from the system. Okay, so mostly done here. I just want to say bigger picture, why we're excited about this system, how we'd like to leverage these synthetic organelles is for a number of applications in cellular engineering and regenerative medicine. So I've shown you, control of cell cycle, signaling pathways. We're also really interested in targeting transcription factors that regulate cell fate as a modular strategy for pushing stem cells down different paths for renewal and differentiation. And in addition to this utility for the condensate platform as like an insulator, we'd also like to use it in a bioreactor scheme, but not for metabolic engineering, but instead to think about evolving new signaling pathways and responses in cells. And so previously, a number of groups have forced inappropriate interactions among signaling components. In this case, it was through domain recombination and fusion to generate new pathway responses to inputs. And so what I think is that by combinatorially tagging components in mammalian cells, it should be feasible to think about enforcing their co-localization to promote new interactions not normally present in a cell. So using mesoscale condensates, this overcomes issues of titrating components from just using single scaffolds as a way or adapters to promote this cross-reactivity. And I'm excited about this system because I think it's compatible with large-scale epistasis testing in yeast or CRISPR tagging screens in mammalian cells. Okay, so to wrap up, what I've told you about today is our ability to characterize from the bottom up a novel protein condensate platform that we can deploy in living cells. Some of the keys were figuring out ways to get highly efficient partitioning by lowering CSAT. We're able to functionally knock down components, we insulate them from their pathways by targeting them using these coiled coils to our synthetic condensates. This is really a modular strategy. So in theory, excluding some integrated membrane proteins, we think that many diffusible proteins inside the cell could be sequestered as a way to, we show regulate polarization, proliferation, and cytoskeletal spatial organization. And there are a bunch of great condensate systems that have demonstrated, optical regulation. In this case, because we start with such a strong amount of sequestration, we start in the inhibited state, and we demonstrate this reversible or irreversible switching by controlled client release, which I think is a really useful strategy if you want to quickly, release huge amounts of component into the cytoplasm or nucleoplasm to modulate cell response. Okay, so I want to wrap up by acknowledging a number of people, including members of my lab, Mikhail and Wentao, who really carried out a huge portion of this work. I also like to thank collaborators. I really want to acknowledge Dan Hammer and Jiten Mittal, who we have long-running collaborations with on designing condensates, and Jiten particularly for the computational work. And I want to acknowledge generous funding from various sources and you know, the opportunity to speak here. So I'm happy to take questions at this time.
Thank you very much, Matt. There is a whole bunch of questions in chat for you.
Okay.
Can you see chat or?
Yeah.
Okay. Yeah, if you would like to read the question before you answer it, that's going to help to people watching the video.
Yeah, I'll start from the top. So it looks like the first question is from Pat Sievert. That's, would it be possible to use this condensate platform to create a pseudo eukarya to create artificial organelles like the mitochondria and others in bacteria? Yeah, so this is a great origin of life question. I think any kind of coacervate system in vitro is, you know, there's a number of labs thinking about how to use this to, you know, as a starting point for concentrating biochemical components and self-replication. So I guess the short answer is yes, but I don't know how to do it. But I think it's a really exciting idea. All right, let me go to the next question. Are these condensate proteins easy to express, or does it take inducible expression and tweaking? Yeah, this is a good question. So where we started with actually was work from Jim Shorter and Aaron Gittler, demonstrating expression of a variety of types of proteins that aggregate in budding yeast. And one of the problems is if you have constitutive expression and these condensates happen to be toxic, then you run into a problem where the cells you'll generally select fastest growing cells that have the lowest level of expression. So that's why we use inducible expression. You know, we can tweak parameters in the system without having to think about growth. Now, since then, we have realized that this triple RGG with the TLPA tag has absolutely no effect on cell growth at all. So in fact, you can constitutively express it. But the reason that I would just generally promote some type of an inducible expression system is that any kind of modification that you do, even if it creates like a 2% change in cell growth rates, you'll have to, you know, you'll eventually select for lower expression. Okay, next question is from Charlotte. So can you use antibodies to facilitate client recruitment instead of CRISPR-based approaches? What else is recruited to your synthetic condensates? Are you concerned that other aggregation-prone proteins, right? So this is an orthogonality question. So maybe I'll try to answer the antibody question, which is if you're thinking about inside a cell expressing like a Fab fragment or a nanobody as a way to bind to a ligand or kind of an active site of a protein as a functional blocking strategy, that of course could work. In this case, we're not just functionally blocking a protein, but we're also subcellularly anchoring that into our synthetic organelle. And so I think really for proteins like the GEF-CDC24, which has to localize to the plasma membrane to act on the GTPase CDC42, this additional element of having it clustered in a condensate, physically segregated from its compartment, adds an additional layer of efficacy for knockdown. But certainly there are a number of strategies, you know, even anchors away for segregating components inside a cell to insulate them. And in terms of other aggregation-prone proteins, so this is a good question. We've looked at whether other IDRs will co-segregate with RGG condensates, and indeed we see a number of them that do, although a lot of times it's through other RGG domains. So like FUS full length has an RGG domain. It does segregate with these synthetic RGG condensates, but if we just look at FUS-LC, for example, it will not. But yes, this is certainly something we're not, we essentially use cell growth initially as a test of orthogonality, but I think in each pathway you'd want to target, you know, it's worth considering, you know, what controls would be necessary to really demonstrate that you don't have some other detrimental non-specific recruitment of RNAs or proteins. Can you express these in cell-free systems? So absolutely. It's really a question of expression level above the critical concentration. And so for that RGG triple, it's basically in the range of 100 nanomolar. So if you can get cell-free expression higher than 100 nanomolar, then you can generate condensates from these transcription translation systems. We haven't directly tried that ourselves, but we've definitely thought about it. Could these artificial organelles be used to control activities of synthetic artificial cells? So absolutely. Actually, our original characterization of these components was inside water and oil emulsions. We since also characterized them in unpublished work in polymersomes and in dendromersomes. And so, we think about them as a hub for controlling information flow, but also, if you want to pre-sequester a component of interest and then release it on demand, certainly that was one of the reasons we wanted to work with this platform, is that we could do this characterization ex vivo and in these synthetic cell-like systems. Next question from Denise Berkeley. Can you just encapsulate this coacervate within a liquid? Can we use a coacervate with a membrane on its own without a cell? This might be, I'm not sure I totally understand this question. I'll try to give two answers, which may not be what you're actually asking about. But one, you can make these in vitro. They're stable. They will fuse, but if you have them just on a cover slip, then you could, study them in isolation. And if you're asking about putting another membrane around them, we haven't done that, but we have been able to localize proteins to the surface of them to create some kind of a skin or a shell. And I could talk to you more about that. Ava Davenport, another question about origin of life, astrobiology, people on the call here. You could potentially have awesome collaboration with that community. Great, happy to chat more about it. Question from Peter, did you establish the size limit to payloads you could attach to the coacervating protein? So what about large soluble sequence specific RNA binding proteins? Yeah, so this, fundamentally, we know something about size limits, but what I will tell you is that the ratio of folded to disordered protein seems to be really important. And so once a cargo is much, much larger than the size of the scaffolds, then it starts to affect, so it generally increases the CSAT. And so it reduces the fraction of total scaffolding client that then gets inside the condensate. Now you can fight that with expression. The largest protein that we've targeted inside one of these is 110 kilodaltons, and that was quite efficient. Hopefully that gives you some idea of size limits, but we haven't, for example, tried to target Titan or other really large proteins. Again, we think that there are going to be rules to the ratio of folded to IDR for partition coefficient. So expressing these proteins in a cell-free system, we're happy to share constructs. We have ones for bacterial expression, yeast expression, and mammalian expression. So, we're happy to provide those. Emma, thanks for the question. Whoops. And the last question, Denise, yes, I mean, putting another membrane around them. So we haven't directly tested this. We can encapsulate these inside of bilayer vesicle compartments, and they don't necessarily associate with the membrane. Right now, I think the easiest way to think about a surface skin is a protein-based amphiphile, which is how we've approached the problem. But I'm definitely open to thinking about, how to get, lipid amphiphiles to interact with them. I think that's all the questions.
Yeah, I think that's all the questions. Yeah, I don't have any other questions in chat. Oh, one just popped out.
Okay, so targeting. Have you tried to target the proteins to the ER? Luis, maybe you can clarify slightly. Do you mean the client in terms of kind of an anchors away strategy of targeting it to the ER, or the scaffold itself and the condensates to assemble these condensates inside the oxidizing environment of the ER lumen. Maybe you can clarify. So yeah, no, we have not assembled these condensates inside of the ER. That would be an interesting point. Add a signal sequence and you know, figure out these are free of cysteines, so that's potentially useful. They will not be cross-linked. That would make them gel-like, I think. But yeah, that's something we could definitely try. We haven't done it. Follow-up question from Charlotte. Right, so a lot of questions about localization using an NLS or an NES. One of the reasons we really like the LAF1 RGG domain is it's strongly cytosolic. It almost never ends up in the nucleus, or even if you co-express it with another IDR that goes to like the vacuole least, the RGG domain is really quite orthogonal and stays cytosolic. But we've also looked at FUS-LC. This is a really common IDR that gets used for building these condensate systems. And the FUS-LC sequence is strongly nuclear, and particularly when you multimerize it, it becomes extremely nuclear. And so we have to use a variety of nuclear export signals to force it into the cytosol. But the short answer is yes, you can add an NLS to the RGG sequence and get it inside the nucleus. You can add an NES sequence to the FUS-LC domain, get it into the cytosol. So in theory, we can generate these condensates in different subcompartments inside the cell.
Thank you. So I want to piggyback on the localization questions. Could you use it to anchor somehow mRNA and do local translation? I'm especially asking from the point of view of looking at the local translation of synapses.
Yeah. So the Lemke lab has done something like this in mammalian cells. They wanted to optimize non-natural amino acid incorporation. And so they figured out a strategy to essentially get the tRNA transferase components inside condensates in the cell and accelerate incorporation of a non-natural amino acid. So yeah, I think with an RNA binding protein of interest as a client, you could drag in potential RNAs. And for localization inside the cell, you know, generally where we would start with would be to add a, you know, signal sequence or targeting sequence to bring it to the subcellular region of interest in the nucleus, subregion of the nucleus, et cetera.
Yeah, that would be really cool.
Yeah, we should talk more about that.
Okay, yeah, I'm assuming your proteins won't just have the neurons turn belly up, because if they are toxic for expression, if you want to do it in primary neurons, that might be hard.
So we have not tried expressing these in neurons. We've tried 3 mammalian cell types, you know, common culture cell types,
yeah,
and they do not. The RGG triple with the TLPA tag does not impact growth rates, but you know, there's that necessary starting point for each system of just ensuring full orthogonality. I don't even want to say full orthogonality, but minimum orthogonality to cell growth. Yeah.
Yeah, and especially with neurons that are so fragile, but that would be really interesting to see if that could be used to do something like changing plasticity of synapses by targeting a certain protein to express their, especially if you have it light-inducible. Although I probably would just kill cells. I don't know, everything kills those cells. So, all right.
Yeah.
I think that was all the questions. That's all the questions I had. All the questions in chat.
Great. Yeah. Thanks for such an engaged audience. This is fantastic.
Thank you so much for giving a talk, and thanks everyone else. And see you all next week.
Okay. Bye.
Bye.